Hello everyone, welcome to our channel Fox Science. Today we will learn about the structure and function of ribosome and the translation initiation in both prokaryotes and eukaryotes. So let's get started. So basically ribosome is made up of two subunit, one small subunit and another one is larger subunit. And two subunit is made up of some ribosomal protein and some rRNA. As prokaryotes and eukaryotes have two different type of ribosome. So generally prokaryotes have 70S type of ribosome and eukaryotes have 80S type of ribosome, and ribosome plays a crucial role in translation process. Ribosome, actually we can say, like, ribosome is the main uh, engine for the translation process. So first we will learn how is the ribosome look like, and how many gates or channels are present in a ribosome. So here in this slide you can see this ribosome is made up of two subunit. One is bigger and one is smaller. And as you can see here, there are three gates are present. Okay, E, P and A. A stands for amino acid site, P is peptidyl site, and E for exit site. Here, in many competitive exams, question come that P site and A sites are present in which subunit. Actually, P and A is present in the small subunit and E present in the larger subunit. So basically the larger subunit contains only one channel or one gate, that is the exit gate. Rest of the two gates like peptidyl site and A, this two gate contains the smaller subunit. Okay, and in the smaller subunit there is one another channel, that is the mRNA binding site. In A site, when the tRNA get attached with amino acid, as we already discussed about tRNA and tRNA charging in our last video. So when tRNA get charged with some amino acid, they come to the A site, means this amino acid site or accepting site. Then the P site already contains the previous synthesized polypeptide chain. Then one peptide bond form in between the new come amino acid and the old polypeptide chain. And when everything is finished, then the vacant tRNA exit through this E, means exit site. So basically this is the basic funda of ribosome and the basic structure of ribosome. Now we will see that how 70 ribosome is made up of what, and 80s ribosome is made up of what. So 70s ribosome basically composed of two subunit, 50s subunit and 30s subunit. So if you compile like 50 plus 30, so that is not actually 70. Then how, why we called it as 70? And in case of 80 ribosome, 80s ribosome also, you can see that 60 plus 40 is not 80. Then why we called as 70s and 80s? Basically, when the single subunit, for the weight of single unit, we calculated separately. Actually, first the bigger or larger subunit and the smaller subunit we separate. Then we did centrifugation, and after centrifugation we measured the weight. So when we measure separately, then we get to know that 70 ribosome is made up of two subunit. The larger subunit weight is like 50s and the smaller one weight is 30s. But when we together, means if we try to measure the intact weight of an intact ribosome, that becomes 70s actually, because some ribosomal RNA or some proteins get precipitated during centrifugation process. So in that case, we are not able to measure the entire weight of our ribosome like that. So same goes for 80s ribosome also. So now you can see here that 50S subunit is composed of 23S rRNA, 5S rRNA and 31 proteins. And the 30S subunit composed of 16S rRNA only and 21 ribosomal protein. On the other hand, you can see 60S subunit is composed of 28S rRNA, 5S rRNA and 5.8S rRNA, and 40S subunit is composed of 18S rRNA. These are very important and MCQ type question comes from this side, that 70S ribosome is composed of which rRNA, 80S ribosome is composed of which type of rRNA. Like that, very in so many competitive exams of life science, you will get to see like this type of question. So, as we already know that two types of ribosomal structure and function, now we will start about the translation initiation, the first phase of translation process. So, it is little bit different in prokaryote and eukaryote. Basically, in prokaryote, some initiation factors are involved. Three types of initiation factor mainly involved in prokaryotic initiation. What are they? IF1, IF2 and IF3, that is initiation factor 1, initiation factor 2 and initiation factor 3. The function of initiation factor 1 is it is basically helping in the dissociation of the larger and smaller subunit. It gets attached with the 30th subunit and then it causes the dissociation of the larger and smaller subunit, and IF2 direct the initiated tRNA to the P site. As I have already mentioned, there are three sites EPA, and this is very informative point, that is the first tRNA or initiated tRNA always come to the P site. Then from the second tRNA used to come in the A side, then peptide bond formation happen. Like that, polypeptide chain synthesis happen. We will discuss it in elongation process. So who bring the first tRNA towards the P side, that is the IF2, or this factor brings the initiated tRNA to the P side. Next, the IF3 or initiation factor 3 is the main important protein in case of prokaryotic initiation process. This actually inhibits the premature reassociation of ribosome, means it always get attached with the 30s subunit and then inhibits the interaction or 50s RNA or 50s subunit to the ribosome, and then it helps in the mRNA and ribosome association. Then it again selects the initiated tRNA also, that which tRNA will act as initiated tRNA, that also IF3 selects. So if there is any mutation in IF3 binding region, so there will be no initiation of translation, means the translation will completely stopped if there is any mutation in case of IF3. So in this slide, you will get a very clear overview for the prokaryotic initiation process. As you can see here, that this is the ribosomal subunit, smaller one, that is the 30S, and this is the mRNA, okay? And this is the A side, this is the P side. So first you can see that in the A side, IF1 get attached and it causes the dissociation of the 50S and 30S subunit, okay? The first step. Second, the IF3 come and sit in this region, and it what it did used to do, it actually prevent the reassociation, means premature reassociation of the 50s. Actually there are so many factors involved before starting translation process, okay? So IF3 sit in this position until everything is finished and the ribosome become completely ready to start the translation process. Until then, IF3 used to sit in this place and block the premature reassociation with the 50s subunit. And in the P site, you can see this is the AUG or start codon is present. So actually the ribosome go and sit there, the 30th subunit sit there where the start codon is present. Then the IF2 bring the first initiated tRNA, and this initiated tRNA gets bind with the P region. So as I have already told that the first tRNA will always come and sit on the P region. And see here IF1 still is in the A region and block the A site, so that the initiated tRNA by mistake don't sit in the A region. It have to come to the P region. That's why the A site also blocked by IF1 or initiator factor 1. Then what happens? Then when the entire complex formed, like the initiated tRNA, if tRNA comes and bind with the AUG start codon at the P region, and then the IF3 gets dissociated. Then when IF3 get dissociated, the 50S ribosome come and sit, and now whole ribosome mRNA along with the first tRNA with the first amino acid, the entire complex is formed. Now the initiation is completed. Then after this the elongation process will start. So in this way the entire prokaryotic initiation of translation happens. So it basically, if I tell in concise manner, so it will be three factors: the initiation factor 1, 2 and 3 conduct the entire process. Initiation factor 1 dissociate the ribosome. Initiation factor 3 sit on the 30th subunit and inhibit the premature deassociation with the larger subunit. Then IF2 brings the charged initiator tRNA to the P site, and after that every initiation factor gets discharged from the complex, and the ribosome become 50s 30s the whole entire complex. And this is how the initiation in prokaryote finish, and then elongation will start. So elongation we will learn in the our next video. Now we will discuss about the eukaryotic initiation process. So as eukaryotes are higher organisms, so there are in so many proteins involved in this process. So it's actually very easy, but the number of proteins in case of initiation is more than the prokaryotes. Okay, so here you can see first this is the 5 prime to 3 prime Kozak sequence ACC AUGG. First the ribosome will search for this region. Okay, and when it will get the Kozak sequence, then there will be two type of translation initiation. One is 5 prime cap dependent and another one is cap independent. Okay, as you already know that in case of eukaryotes the mRNA is always 5 in the 5 prime region, it is always capped, and in the 3 prime region there is poly A tail present. Okay, so in case of eukaryotes the translation initiation can be two type: cap dependent and cap independent. So let's discuss about the factors which are involved in this process. First, EIF3. Why E? E means eukaryote, okay? And same like IF3. What EIF3 do? EIF3 prevent the premature deassociation between 40S and 60S subunit. Then the second factor who come in the scene, that is the EIF4F complex. This is a trimeric complex. Okay, we remember very carefully, many questions come from this side. EIF4F is a trimeric complex, heterotrimeric complex. Three type of monomers are present here. First is EIF4G, which is a scaffold protein. Second is EIF4E, which helps in the rate limiting cap. This is a rate limiting cap binding protein, and this protein actually find the 5 prime cap region and bind with it. Okay, and the third monomer is EIF4A. This is an ATP dependent helicase, and it causes the unwinding of the supercoil structure of mRNA to open the codon. to the ribosome and tRNA. Okay, so these three monomer EIF4G, EIF4E and EIF4A mainly compose the EIF4F complex. Okay, then the next protein come, that is the EIF4B. This is actually helps in EIF4A helicase activity. Then the next protein is EIF2. What it do? Actually, it directs the initiator tRNA to P site. Same like you are getting the similarity between the prokaryote and eukaryote. So here also EIF2 direct the initiator tRNA to get bind with the P site. Now there are two more type of protein, that is EIF2B and EIF5. EIF2B causes the EIF2 activation. Actually this is GEF, means guanine exchange factor. When EIF2 need to be activated, then EIF2 is getting attached with GTP, and when it become deactivated, then it is attached with GDP. So this GTP to GDP conversion is done by EIF2B, as it is a GEF or guanine exchange factor. Then EIF5A and EIF5, there are two other proteins. So EIF5A actually GTP is activating protein. It induces the GTP hydrolysis by EIF2, and then after entire process the EIF5 actually helps the 60S subunit to get attached with the 40S and making the entire ribosome complex. Okay, so there are one more interesting thing, that is the ternary complex. What is this? When EIF2 becomes activated by binding with GTP, then EIF2 brings the initiator tRNA. You can see here, as it is in eukaryote, that is no formylin, no formyl group. So tRNA I mate only, not tRNA F mate. Okay. So here this is called as ternary complex. When it gets attached with the 40S subunit, then we will call it as 43S complex. So question comes here from here very frequently, that what is ternary complex? Ternary complex is nothing but it is composed of EIF2, GTP and the initiator tRNA. This is the ternary complex. When it is get attached with the 40S subunit, then 43S pre-initiation complex is produced, and then with the 43S complex, EIF5 and EIF5A getting attached, and 48S complex is produced. Sorry, I forgot to mention, in case of 43S complex, there will be the EIF2 GTP tRNA imate, the 40S subunit, as well as the mRNA, that is we called as 43S pre-initiation complex. After that, the 48S complex formed, and then the 60S subunit come and bind, and then the initiation process is finished, and then the elongation will start. So when you will watch this video, just note down step by step, it will be clear. So thank you for watching this video. If you have any doubt regarding this, please do comment in our comment section. If you like this video, please do like and share. Thank you so much.